What's up, everybody? Mr. Lanes here. I have my camera taped to my ceiling right now, so this is going to be fun. I'm here today, and we are going to model meiosis, all right? So not mitosis, meiosis. And I'm going to start off with this model. We have seen this model many, many times at this point. This is our foam model of DNA, and I wanted to remind you that when we go from DNA to chromosome, what we're doing is we're taking that DNA and we are tightly coiling it around those histone proteins, and it coils and coils and coils until it becomes a visible chromosome. So we are going to start off with 6 chromosomes, all right? These are unreplicated chromosomes. There's 6 chromosomes. They are unreplicated. They are chilling out in our nucleus area here. And what I want you to think about is that there's some similarities here in these chromosomes. In fact, this chromosome and this chromosome, if we look at them, they are the same length and they have a centromere in the same region. In fact, if we look at all of these, we could actually kind of pair them off. Now, they're not connected. These aren't chromatids. What these are going to be called is homologous pairs of chromosomes. I want you to think of it as like a pair of socks or a pair of shoes. You kind of have a left shoe and a right shoe, or a left sock and a right sock. Think about it in those terms. So what this means is that we actually have two copies of every gene. So let's say this chromosome here has the gene for hair color or some trait. There's going to be a second copy of that gene on this homologous chromosome. Now, they could be the same or they could be different. And that's what I want you to focus on. I'm walking you through this process with the intent that you don't need to know every step. What I want you to pay attention to through this process is, how do we increase variation throughout this process? So how do we increase genetic variation? That's what I want you to think about, all right? So here we go. We're going to go into meiosis. This is going to look very similar to mitosis, but we're going to go through it not once, but twice. And our goal for this is very different than mitosis. In mitosis, we wanted to make an identical copy of the same cell. Same genes, same everything, same number of chromosomes. The goal for meiosis is to make eggs and sperm, what we call gametes. So we're going to try to not have the same number of chromosomes. In fact, we want to reduce the number of chromosomes by half. This is important if you think about that. When an egg and a sperm come together, we want to have 46 chromosomes. So that means the egg has to have 23, the sperm has to have 23 to make 46. If we used mitosis to make egg and sperm, every generation we'd be doubling the number of chromosomes, and that would be a really bad day. So let's get started. 2 things we're looking for. We're looking for reducing chromosome number, and we're looking for how are we adding genetic variation. So pay attention. Here we go. Let's rock and roll. So the first thing we need to do is we need to go through S phase. So we need to replicate all of these chromosomes. So I'm going to do that right here by simply adding in the sister chromatids. So now you can see that each of these has gone through replication, and we have sister chromatids of each homologous pair. Again, let's break that word down. Homologous, the prefix homo means same. So that means they have the same genes, although they might be different versions of those genes, something we are going to call an allele. So they might have different versions of the genes on each homologous pair. I used this one earlier as the example. Let's say this is a recipe book. This blue chromosome might have the allele or the gene for chocolate chip cookies, where the red allele or gene on this one might have the recipe for oatmeal raisin cookies. So they're both cookie recipes, but they're making 2 different types of cookie. So here we go. We have gone through S phase and we have our homologous pairs. The next thing, we'd go through prophase. They'd condense into these visible chromosomes. And then we'd go in prophase 1 of meiosis. What we're going to do is these are actually going to form what we kind of consider like a super chromosome. We call it a tetrad, where these homologous pairs get paired together. Think of like a giant centromere that could come and connect these whole chromosomes together. All right? That's what's going to happen. And when they form this tetrad, tetra, again meaning 4 because we have 1, 2, 3, 4 chromatids, so tetrad. When they do this, something very important happens. Note, something very important happens is they actually, what we call crossing over. So this chromatid here on the blue is actually going to exchange pieces with the red. So it's actually going to flippy-floppy some pieces and, whoa. And what it's going to do then is it's going to have not just red and not just blue. It's going to come up with this patterning of red and blue. In fact, this is going to happen on all of them. And sometimes this goes all the way, you know, like whole chunks of chromosomes get split. Like this one's going to flip this whole thing here. So we have this idea of crossing over. Now, if we look at these chromosomes, I'm going to flip this guy here. This one, wow, this is exciting stuff. And so now what we look at, if we just kind of give it a glance of what's happening, we have these tetrads, so the homologous pairs are together, but now we have very different chromosomes. So we don't have sister chromatids that are identical like mitosis. What we have now is different combinations of genes on each homologous pair. Okay? So that is one way we add variation. We have crossing over. We are changing out the genes and making new combination of genes that have never existed ever before. And this is important because you might be asking, like, well, why do we have a homologous pair? Well, where did these come from? This original red homologous pair and this blue, this one came from mom, this one came from dad. So there's a real danger that when you make your own egg or sperm, you're only passing on the information from mom or dad. So crossing over ensures that you don't just pass on genes from mom or dad, that you're passing on a new combination, that you're mixing things up a little bit before you go on. So that is during prophase 1. What happens during prophase 1 is these tetrads line up at the center of the cell. This is going to sound very similar to metaphase. That's exactly what it's called, is metaphase 1. But instead of separating sister chromatids, we are going to separate entire chromosomes. So we're separating homologous pairs of chromosomes. I'm now going to kind of push this side off, but they're going to be going through the same thing that we're going to go through with these three. Then we go through meiosis II. One last thing, let me back up a moment. During anaphase 1 and meiosis 1, they line up, how they line up is completely random. That's another important part. That's how we add variation. That doesn't mean like all the dad chromosomes are on this one, all the mom chromosomes are there. How they line up, that's important. It's called independent assortment. We're going to come back and talk about that in a little bit. Now, back to meiosis II. Meiosis II, functionally exactly the same as mitosis. Sister chromatids line up, and we separate those sister chromatids. Now this becomes two separate cells. You can think of a nucleus around here, a nucleus around here. That same thing, I'm going to scoot these guys, scooch them over like that. I'm going to scooch them over like that. And then I'm going to bring back these bad boys. And remember, they went through the same process, so I'm going to separate them into their respective nuclei too. And if we look, what we can see now is that we have 1, 2, 3, 4 chromosomes, or sorry, 4 new cells. Each new cell only has 3 chromosomes. Remember our original cell, it started with 6 chromosomes. So now we've reduced the number from 6 to 3. If we look at the genetic makeup of each new nucleus or each new cell, we see that each one has a different combination of genes. That this is very different than this one. That this is different than that. That they're all a little bit different. They have new combinations. Each one of these cells will then go on to become an egg or a sperm. Okay? So that, let's pretend that this is the egg and this is the sperm. When the sperm fertilizes the egg, those homologous pairs, we now return to our original chromosome number of 1, 2, 3, 4, 5, 6. So now we can start this cycle again. Now we have an egg and a sperm coming together to make a zygote, And now this can go through mitosis and do everything it needs to do to grow and develop until it can make eggs and sperm on its own. So again, in summary, we started off with one 6-chromosome cell. They crossed over during prophase to make these combinations that had not been seen before. Meiosis I, we separated those homologous pairs. The way that they separated in Meiosis I is totally random, adds more variation. Meiosis II, exactly like mitosis, making those 4 daughter cells that are genetically unique, and they go on to make the egg and the sperm. That is the gist of meiosis. That is what I want you to know. Again, how do we add variation? Crossing over, independent assortment, that's important for right now. We're going to come back and talk about some other things later. I hope this was useful. We'll talk to you soon. What's up, buddy?
I want to see the video that you took.
I do too.